Hello, my name is Scott Badish. I am the President and CEO of the Autism Society of America, and it's our delight and honor to co-host this wonderful webinar called The Therapeutic Promise of CBD on Autism with the Wholistic Research and Education Foundation, along with the University of California at San Diego. I know there's a tremendous amount of people listening to this webinar, and one thing I want to start, it will be about an hour. It won't last more than an hour. Secondly, if you have questions on your page in your computer under the chat, please put those questions. Inevitably, with so many people, we will not be able to answer all the questions tonight, but during this webinar. But for those that we can't, we will get back to you with the answers. I want to just say how the Autism Society of America, for those who don't know, is the oldest and largest grassroots autism agency in America. Our job is to help people maximize their quality of life, and when we partnered with the Wholistic Research and Education Foundation about CBD oil, we did it for a couple reasons. We know a number of people call us and call our call center daily wanting to know about the benefits of CBD or medical marijuana, and there's a lot of confusion out there, and we thought this would be a wonderful way to help address that confusion. And secondly, we want to help people maximize their quality of life, and if someone could make an educational decision for what's best for them or what's best for their child, we want to be there. We also want to say the Autism Society of America does not have a position on a medical marijuana or CBD oil. As you'll see from this webinar, we're working to see best practices and proven research to back up things. And we also encourage that anytime you use anything that relates to treating a person with anything, that you go to your doctors and have your physicians know about it, and hopefully this will give them research. This will be on tape, so if they want to see it, they can see it. I also want to thank the University of California, San Diego for putting on this webinar, for filming it, and thank you very much. And it's my pleasure, someone we've worked with who's just been a very wonderful person, is Palin Thorogood, who is the co-founder and president of the Wholistic Research and Education Foundation, which is the real emphasis of studying the relationship of CBD and autism. Palin, it's my pleasure to introduce you. Thank you.
Welcome. We're delighted that you have joined us today to learn just what kind of therapeutic promise CBD holds on autism. We have some brilliant minds here with us today who have been studying this very important topic from many angles. And they'll share with us their insights to date, as well as talk about what they hope to learn from some groundbreaking research that is already underway here at UCSD. But before I introduce my esteemed colleagues, I'd like to tell you a little bit about our foundation, as well as about CBD, which is fast becoming a miracle medicine of sorts. And hopefully with this information, as well as the insights that you will gain from our doctors and researchers on the panel, you can separate fact from fiction. Wholistic Research and Education Foundation is a California public charity that is dedicated to exploring the health benefits of CBD-rich therapeutics across a variety of conditions, including autism, and advocating for safe and legal access to those in need via education and advocacy. Our desire is to understand if, how, and why CBD may benefit certain conditions for certain people, which is why we fund only multidisciplinary research that brings together clinical, scientific, genetic, neuroscience, and neuroimaging disciplines, just to name a few, for a systematic and comprehensive view into the underlying mechanisms at play. We want both the public and the healthcare professionals to be empowered with the latest scientific data, which is why we're committed to education and sharing this information far and wide, including venues such as this. We also believe that legal access to products that are deemed safe is critical. Given CBD and other cannabis-based products are becoming so prevalent across much of our country, which is why, once again, we leverage this scientific information to advocate at the state and federal level to increase access to all families in need and provide better treatment options. So, what exactly is CBD? CBD is the non-psychoactive compound found in cannabis, along with 150 or so other cannabinoids found in the plant. Numerous studies show that CBD has anti-inflammatory, antioxidative, neuroprotectant, and anti-inflammatory properties, which is likely why CBD has been shown to be effective across so many health conditions. This is probably a good place to also tell you that CBD can come from both the hemp and the marijuana plants, both variations of cannabis. Over the years, marijuana has been bred to include a higher concentration of the psychoactive THC compound, whereas hemp has been bred for exactly the opposite purpose. Legally, hemp has less than 0.3% THC. So with the introduction of the historic 2018 Farm Bill, hemp is going to be able to be grown legally across all 50 states, and CBD derived from hemp will be removed from the Controlled Substances Act. This should make hemp-based CBD products much easier to access and will also have better regulations about the safety and the efficacy of these products. So cultivation, manufacturing, distribution, and access all should become a lot easier for these wellness products. Also, it's important to note that the WHO, or the World Health Organization, has published a very comprehensive report on CBD last year, November of 2017, which deems CBD safe and effective both for humans and for animals, and it indicates that CBD poses no public health risk. The WHO indicates that CBD has been very effective for epilepsy, which is likely where most of us have heard about CBD. And FDA recently also approved a CBD-based product, a drug for the treatment of epilepsy. The WHO further states that preliminary evidence indicates that CBD can be helpful for numerous other serious health conditions, including cancer, Parkinson's, Alzheimer's, and psychosis. So, how does CBD work? CBD works by interacting with a major biological system called the endocannabinoid system, or ECS. ECS was discovered in the late 1980s, so relatively recently, and it is found in all mammals. It has the single largest receptor network in our bodies, and it can affect almost all cells, muscles, organs by regulating nearly all major systems in our body, including the nervous system, endocrine, immune, and digestive systems, even our heart and our brain. This is why CBD has been shown to have effect on so many serious conditions. We've already talked about its effect on epilepsy and how it's becoming approved to be used even through the FDA. CBD has also been quite effective with anxiety and depression, including PTSD. It can help with inflammatory disorders such as arthritis or Crohn's disease. It can help with neurodegenerative disorders such as Parkinson's and Alzheimer's, neurodevelopmental disorders such as autism. It may even help with cancer. However, we just don't know enough as to how it helps some of these conditions and why it's more effective with some people and not with others. It is really this question that has been part of our mission to understand the mechanisms at play, how CBD works, why it works, which is why we are funding scientific, and clinical research to really understand the underlying mechanisms at play. Now, it is with great pleasure that I introduce you Dr. Keith Vaux, renowned pediatrician and geneticist who has been working with autistic children for over 2 decades, and in many cases has recommended CBD when he believed it would help the child or to help the patient in need. He will give you a brief history of medicinal CBD, as well as share from his vast experience. So please join me in welcoming Dr. Keith Vaux. Thanks so much.
Thank you. Thank you. It's a real pleasure to be here, and I appreciate being on this panel. So I'm just starting off. I take most of my inspiration from my dog. So when things, get bad and you feel really tired and, that head's getting heavy, you just find a place to prop it and keep moving. So I have a foundation as well that I started. I was at UCSD for quite a bit of my career. I am a pediatrician, and I trained as a dysmorphologist, but got into sequencing, and I've been working on sequencing for quite a while. The fascinating thing for me was that once we started sequencing, we were finding many, many associated genes, and the question always was, well, now what do I do? And I really found it to be a very in-depth and very troubling question because I didn't really have an answer. Certainly using neuroactive drugs are what we do for the most part. Those drugs, to me, sap a lot of the life out of the patient, and in particular, patients that have autism who have fulfilled every inch of my life. I have a family member that's on the spectrum who's done great, and so to see what the treatments we have do to somebody was really heartbreaking to me. So it really got me motivated to want to look for alternatives and other ways to explore what's happening. So I started a foundation with the help of a couple of my friends, and we focus mainly on exploring novel treatments. So this is one of my favorite quotes, and I've actually probably done most of my career this way, probably to the detriment of my working for corporate medicine, but anyway, it's another story. So this is from Thomas Kuhn. Science is not a steady cumulative process, but a series of peaceful interludes, which is what we've been in, punctuated by intellectually violent revolutions, which is where I think we're heading with autism treatment. I think that the standard therapy is doing a disservice to everybody, and I think we're looking at a paradigm shift coming up, and I would like to be part of that. So I went over a little bit of the history first because I think it's important to remember that we practice medicine. We don't know medicine, we practice medicine. So you always have to be learning, you always have to be exploring, but you also need to understand the giants that came before you and know that they knew a lot of stuff that we've forgotten and that we need to relearn. One of my great friends is out in the audience, and we're learning so much through the work that she's done about diet and nutrition. I mean, how many times has your doctor talked to you about diet and nutrition? Never. We forgot that. We forgot how important that is. And so I'm bringing this up because cannabis has been around and cultivated by humans as long as we have knowledge of what has happened to us. So, you can go anywhere from 2,000 to 4,000 years and see that there was cannabis involved in holistic treatments and faith healing and all kinds of different activities. It appeared in one of the most amazing books probably ever written about medicine by a Persian in the 900s, which basically cataloged every known substance that was used in healing at the time. It's an incredible read if you ever have a chance to look at it. And then in the 1830s, an Irish physician actually brought cannabis to the West, which was Europe at the time, and used it for treatment. And it actually got to the point where the end of the 1800s and the beginning of the 1900s, cannabis was a regularly prescribed treatment. So was opium, and so was willow bark. Those were kind of the big guys that everybody had. This is a photo that shows each, you can't really read them, but this is what the doctors carried around as they went from house to house treating their patients. So you had opium, you had willow bark, which is aspirin, and you had cannabis, and that took care of your anxiety and your stomach pain, some of your gastrointestinal complaints. This is well published. It's all over the literature from that time, but we've forgotten it. I also probably have that same bag. I think that hasn't changed a bit. I don't know if I put my computer in it now, but. So this is when the big change started. And for me, and for all of us that practice, we sometimes feel like drug pushers, but we push them basically on a result of what happened here. So salicylic acid was isolated from willow bark, and in 1899, Bayer patented it. They put an acetyl body on the edge of the salicylic acid, and then that became a patentable item, and that was aspirin. And that's been the first real blockbuster drug. It's still the biggest selling drug in the world. They don't make any money off it, but it's still huge. Again, as I mentioned, opium and marijuana also came into use right around the same time. But what happened was the opium use became so common that the physicians stopped using it. There was a lot of pressure to move into these other manufactured drugs. Again, something we're seeing right now, again, as history tends to repeat itself. So at the turn of the century, about 1 in 200 U.S. citizens were addicted to opium at that time. We're not quite there, but we're getting there quick. So the government started to step in and regulate what was prescribable by physicians. And there was the Narcotic Act in 1914, and then marijuana got into it at that point too. What happened with the Narcotic Act was it was an anti-opium act, and then marijuana became the go-to drug. So it was sort of perceived as not quite as classy as opium, so they quickly regulated that. It was officially banned in 1970, and then again banned even harder during the Anti-Drug Abuse Act and with the Reagans, both in 1986 and 88. It became an offense that was jailable at that point. Prior to this, it wasn't usually something you got arrested for. But what was interesting for us is that then it became illegal to prescribe it as well, and with the accompanying jail time. And as we tend to do, California and Oregon, two of the more progressive states legalized medical use despite the federal regulations, California in 1995. And again, this is an important background because from 2001 to 2010, there were 8.2 million marijuana arrests in the U.S., and that's 50% all drug arrests. That's just crazy. So when you try to get your doctor to talk about marijuana, this is the background that they're using. Now, we've gotten to the point where this year there are now 46 states that allow medical use of marijuana and many states that allow recreational use. The problem is that we're still licensed federally, and this is still a Class 1 medication. Therefore, we cannot ever prescribe it. It's illegal for us to prescribe it. There's a workaround, which is recommending it, but again, in most states, if the federal government decided to change what they were doing, we would all be prosecutable if we ever wrote it as a prescription. Now, that's the way I feel about this, because my patients, the patients that I spend each and every day caring for and up at night worrying about, are taking a substance that I can't talk to them about. And so I feel that I have all this good knowledge about cannabis, but I can't say anything. So I'm just banging my head against the chalkboard. But I see patients, even today, I have a young man who's 15. He suffered epilepsy since he was born. I've had him for about 2 years now. He was on a very, very, very strong regimen of psychoactive medications, prescribable medications, and he couldn't do anything. He couldn't read. He just sat in his bed and did nothing. So we started to wean those medications, and the recommendation using one of the naturopathic physicians that I've collaborated with was to go to CBD, 20-to-1 CBD THC, which is one similar to Charlotte's Web, which I'll talk about just briefly. I saw him today. Despite having roseola, he did not have any episodes of seizures on only CBD. Now, in the past, when he got fevers, he would have large series of epilepsy events and would usually stop breathing and would end up in the hospital. We were able to get him home last night just by giving him an extra dose of CBD. So, again, as a scientist, I don't want to hear myself say, so see, it works, but in his case, it certainly was much more beneficial to him than the medications he was on before. It's also important when you are trying to communicate with your physician that you understand what the terms are. I just briefly went through this. Palin explained this briefly, but there's the cannabis plant itself. And the two main types are the first two on there, and there's a third one that's coming into action now. There's growers and cultivators who are good, and growers and cultivators who are not so good. So it's very important that you understand what the plants are. Marijuana itself is the dried cannabis plant. So it includes the leaves, the flowers, the stems, and the seeds. That's different than hemp. Hemp does also have CBD in it, but there'll be more on that at a later point. So cannabis has, right now, we understand about 400 substances in it, and somewhere in the 100 to 150 of the substances are cannabinoid-type substances. The ones that you've all heard of and the ones that are most studied are the delta-9 tetrahydrocannabinol, which is THC, and that's the neuroactive portion of it. The cannabidiol, or CBD, works on the pathway that Palin mentioned. And there's two others that are coming into use. There's the D8 THC and the CBN, and both of those you're going to hear a lot about them coming up. The really fascinating thing, and I'm just going to touch on this, is the endocannabinoid system itself. And we're starting to just understand it, and I have a partial understanding of it, but I heard it best described as most of our neurons, for obvious reasons, are active. They're under constant stimulation and constant activity. It makes sense, if the ball's coming at you, you don't want to have to start the engine and wait for things to happen. You want to be able to dodge it. So the endocannabinoid system downregulates that normal activity, and therefore for kids that we deal with, kids with autism, kids with ADHD, kids with intellectual disabilities, that regulation piece is really what's at heart here. So being able to calm yourself down. So again, I sometimes feel like this as well. I'm watching my shows at home and I don't really want to go to work, but we've all seen this. So we've all seen this kind of activity. This young man's having a meltdown. Again, with the active medicines that we have, you don't want to have to give this kid Ativan or something similar to that. It would be nice to have something that would be able to calm down that situation and allow him to function. So how about the data itself? So with epilepsy, the data has been very clear, to the point where the FDA approved a cannabinoid CBD derivative medication that can be prescribed, and that's down at the bottom. But we don't have the same good studies with autism. Part of the problem with that is that there's not a autism. With epilepsy, you have an EEG signature that you can show improvement. With autism, as the saying goes, once you've seen a child with autism, you've seen a child with autism. That makes it very difficult to do scientific studies of any type. However, Israel, as with many things, has led the way in this. They see and recognize that this medication is very powerful. So Dr. Aron, in 2018, published this retrospective review of 60 of his patients. They were taking a very similar one to Charlotte's Web, 20% CBD, 1% THC. They were observed for 7 months. They had some pre-testing and some post-testing, and the results were just astronomical. So they were an 80% decrease in the parent-reported problematic behavior. Now, the one thing that's important to say in this is that these were kids that did not respond to standard neuroactive medications. They were not responders to any of the meds we normally use. So this is even larger with that in mind. There's a 50% increase in communication with the parent by their report. So 40% decrease in self-reported anxiety. And for a lot of us, this is a big point, which is 30% decrease in parental anxiety, which is a major bonus, as you know. Now, this was not a controlled trial. There would not be any one that would be able to say this is scientific proof. It's, the patients essentially are self-selected. But it's a very promising start, and hopefully, with the group here today, we'll be able to make more scientific, definitive statements about this. So again, I want to emphasize that the epilepsy data is clear from a scientific standpoint. It is there. So Charlotte's Web being the model of this particular one, this is a 20% CBD to 1% THC. That seems to make the CBD more functional in this particular setup. As I mentioned, there is an FDA-approved medication for these two very rare epilepsy syndromes, but it's going to be used more in the future. I also just want to briefly touch on Marinol, which has been around forever. This is a synthetic THC. This is used for diet, for stimulation of diet, particularly in cancer patients, and then in some eating disorder patients, it's starting to get used. So this also points to some promise in that area. So that's basically the summary of my talk. I have somewhere north of 200 patients that are actively on the medication. I have had, again, very good results with it from parent reports. But I'm really excited that this project is happening. I think we have a team together that's really going to make a difference here. This is another young gentleman that ended up the family moved to Colorado to get Charlotte's Web for 300 plus seizures per day that the young gentleman was having, and he has none moving forward. This is one of the cultivators. It's become a whole science now. It's like a brewer. They really concoct these plants in an incredible fashion. So I have placed, or I will place this afternoon on my website, some references that I brought up here. There's some fascinating articles on animal studies and THC, but again, we're just moving forward with the autism group. So I appreciate the opportunity to speak to you today, and I look forward to your questions. Thank you.
Before I invite Dr. Allison Miotri, I wanted to dive into two areas that Keith talked about. As I mentioned, CBD can come from two sources, both the hemp plant and the marijuana plant, both variations of the cannabis plant. And as I said, hemp has less than 0.3% THC. Now, with the passing of the 2018 Farm Bill, hemp will be federally legal. And I also want to point out that the UCSD study that we'll be discussing next uses CBD only, though THC itself has many medicinal benefits. And other studies that we're funding for other conditions, we're actually trying a combination of CBD and THC with the researchers to see what the medicinal effects together might be. So there's definitely the whole plant of hemp, the whole plant of cannabis has many medicinal benefits. And while it's important for us to study individual compounds, it's also important for us to look at the whole plant and how it may benefit. Second, I wanted to talk about the Israeli study that Keith talked about as one of his last points. It's wonderful that we were able to see a significant percentage of the autistic children who were treated with CBD we were seeing major improvements in behavioral, communication, reduction in anxiety, etc. But while some kids saw those benefits, other kids did not. It was really the curiosity as to why there was a difference in the kids that fueled our funding of the UCSD research, which is truly a first of its kind multidisciplinary research that not only looks into the safety and efficacy of CBD and autism, but also looks into the mechanisms at play, so we can better understand why some kids benefit while others don't. And hopefully we can also explore if there are any modalities that we can use to be able to improve the odds. So with that, it is my great pleasure to introduce you Dr. Allison Miotri, who is the director of the UCSD Stem Cell Program, as well as one of the 3 PIs or principal investigators in the autism study that we have funded earlier this year. Allison?
Thank you, Paley, and thank you for organizing this meeting. I think this is very helpful for the families and people who are online are curious about the CBD and exactly how it acts in brain cells. And that's my job here, is try to figure out exactly how this molecule that it can be extracted from those plants, how that can affect your behavior in some of these neurological conditions. So, but before I jump into that, I'll summarize, the aims or the goals of these studies. And the first thing is really to determine, using clinical evidence, if CBD is safe, tolerable, and if alleviates some of the symptoms of autism. And we will be talking about a specific type of autism that are the most severe type of autism. These are traditionally more difficult to study because the behavior, the stress on the families to participate on this kind of studies. But we are all making an effort to include the more severe types of autism, and we think that's when the benefit might become more evident. So we're going to be recruiting autistic individuals who have aggressive behavior, hyperactivity, and low level of social communication. So the second goal, or the second aim of these studies, is to determine if the potential mechanism of action of CBD actually relates to the alterations in neurotransmitters or how the neurons communicate to each other, improvements in the neurophysiological normalities that we see in these neuronal networks, and overall, if there is any improvement in brain connectivity. So there are plenty of evidence suggesting that autistic individuals have alterations in these brain communications. So we are designing our study to really prove if CBD can act on that. And I think finally, and perhaps one of unique aims of this is to determine whether there are biomarkers for neuroinflammation that are affected by CBD administration in autistic individuals. So that would be really helpful moving forward to identify the population of autistic children and even adults that might be more susceptible to this kind of treatment. So this is a truly multidisciplinary approach. There are many disciplines that are involved on that. There are clinical and neurophysiological evaluation that would be done before and after treatment. Would do the intervention using synthetic CBD initially in 30 children with a severe type of autism, as I said. The clinical part is going to be led by Doris Trauner, who is also a professor in the neuroscience department at UCSD. Then finally, we move on into the more mechanistic, molecular, cellular analysis of CBD acting on brain cells. To do that, we're taking advantage of a very interesting technology that involves stem cell biology, and this is by reprogramming cells from the subjects that are participating in our study and reprogramming them in the lab, creating what we call induced pluripotent stem cells. These stem cells can be driven to specialize into the brain cells, so we can study the effect of CBD in these brain cells derived from each one of the individuals that are participating in our study. And finally, you're going to hear a little bit later on the analysis of these networks that we can collect it in a very dynamic way, because these are live cells in the lab, and this will be done by Dr. Gabe Silvia here at UCSD. So the clinical study, it's quite simple. It's very similar to many of the clinical studies that perhaps you hear about or are familiar about. So these would be 30 kids with severe type of autism that would be pre-screened in between ages of 8 and 12 years old. It's going to be a randomized crossover trial of CBD versus placebo. So we're going to know exactly, if there is a positive effect on top of placebo here. And both cognitive and behavioral tests would be done before and after treatment. So we're going to be collecting lots of data regarding social behavior, language, all type of stereotypes as well. Also, importantly, we're going to be collecting brainwave activity. This is what you can extract using electroencephalogram or EEG. Also eye tracking, and we're going to be measuring some of these movements, spontaneous movements that individuals with autism will often have. And finally, we're going to have several types of brain scan on resting state for kids with that are part of the trial before and after the treatment. So overall, we're going to be collecting tons of clinical data, and I think the challenge is to collect these, to connect this clinical data with the molecular cellular analysis that would be done in vitro in the lab. So I briefly touch on the idea that we can reprogram cells from all the subjects participating on the trial, and we can induce the cells to differentiate into brain cells. Most of the time, we do that in 3 dimensions, and these cells self-organize in something that we call a brain organoid. Most of the time, you're going to see in the media that they call these mini brains. It's not a very good name because they don't actually look like a mini brain, but it's a cluster of cells that have electrical activity going on, and they self-organize on something that resemble the human brain. And before I move on, I want to make very clear that as any other model to study the human brain, there are limitations. And these are the potential limitations that we anticipate. Most of the time, when we specialize in these neurons in the lab, they are more immature neurons compared to the neurons that you have in the brain. These organoids are not vascularized, not all cell types are represented. We don't even know if the conditions that we are growing these organoids are ideal. And finally, we don't know if what we learn into the tissue culture in vitro can translate to the human brain. So the translational potential of this technology is still unknown. That's why this study is so important. It is the first one that will be able to connect the in vitro data towards the subjects in a clinical trial. So I do think that this technology will be disruptive in the way we understand neurodevelopment, autism, and the impact of CBD on social human behavior. So the way we create these mini brains, we follow a recipe that has been developed in my lab for the past years. We start with these pluripotent reprogrammed cells from each individual. So each one of these cells will capture the entire genome information for each one of the subjects. And through a series of process and media and factors, we guide these cells to self-assemble in 3D floating structures that, again, resemble a very mature human brain. And that's what we call a brain organoid. These brain organoids, they can mature for long periods of time, and we have been collecting data from them. The way they self-organize is quite interesting. In the video here, what you see are some of the progenitor cells that are migrating out of the center of this colony. And as they migrate out, these neuroprogenitor cells will stop, will differentiate, and start forming connections to each other, forming what we call the cortical plate. The cortex in the human brain is the center of most of the sophisticated cognition that we have, and is one of the brain regions affected in autism. So for these particular studies, we're going to be focusing on the cortex. So we can measure the activity of these organoids by plating them on dishes that contains multi-electrode arrays printed in the bottom of the dishes. All the time when there is activity passing through these electrodes, we can capture that and generate an activity map. So we can simulate how the networks are firing, or how the neurons are being active and connecting to each other by studying these kind of networks. And Gabe Silva will tell you more what we can do with this data later on. So very surprisingly, we learned that we can keep these brain organoids for long periods of the time. And as we do that, they age in a way that is very similar to human development, reaching to a point where the activity is super high. For example, in the first graphic here, I have time from 2 months to 10 months, and the number of spikes or activity from those neurons are moving from 3,000 per second to a little bit over 300,000 per second. So there is a time frame where the networks really get organized and become super active. And because we have such an amount of activity, these networks start to generate brain oscillations. And these oscillations is what we can detect it by using EEGs. And so that will be the basis of our comparison with the clinical data. The clinical data, all the subjects will have the EEGs done before and after treatment. And we can use the EEG collected from the subject's brain organoids and be able to compare that with before and after CBD treatment. So virtually what we are doing is running the clinical trial in parallel, in vitro, in the lab, and hoping we can match the data that we have with the clinical data. By doing that, not only we can see who are the responders and non-responders, we can actually search on the genome what are the potential genetic variants that account for these differences. And we can also test many different concentrations and doses of CBD and THC to actually personalize the treatment for each individual. So that would be something closer to a personalized treatment for each one of them. So this is just one example of how the networks changes over time. So I'll stop here and pass to Gabe Silva to talk a little bit more about the system
before
approach.
Dr. Silva speaks, I just wanted you to clarify something for our audience. Where do you get the induced pluripotent stem cells?
Yeah, so the induced pluripotent stem cells will be reprogrammed from a small 3-millimeter skin biopsy from each one of the subjects.
Okay,
so these
so it's
are
coming from the
fibroblasts
skin directly.
from the skin cells. Yes.
Okay, great.
All
Thank
right.
you so much.
Thank you very much. So, and for the opportunity to participate. So just to pick up where Allison left off, I'm a theoretical and computational neuroscientist, and one of the things that we've done is we've essentially developed, so this is sort of the most innovative, kind of far-reaching part of this study. We are going to take Allison's models, the organoids that they're deriving from the individual patients actually participating in the clinical trial, and carry out a sort of very new, rather sophisticated mathematical analysis of how information flows through individual neurons and then through networks of neurons in a dish, essentially, and the types of organoids that Allison was showing. So what we've done is we have a set of a mathematical theory, a set of mathematical equations, essentially, that says something about how signals propagate through a network, how information flows through a network. And what we've been able to identify is that there has to be a balance between the nodes, the internal components in the network, how they're holding on to information as they process that, and then the rate at which, the speed at which information is propagating through the whole network, right? So when that balance breaks down, we've shown in simulations and again sort of mathematically that there's a breakdown in information processing. And we've recently shown that in typical neurons, in normal neurons, this ratio, this balance, which we call the refraction ratio, between this information processing within individual nodes and the speed of information flow within the network, that ratio is an optimization principle that neurons are actually designed, they're optimized for. Their structure, their actual shape is optimized for that ratio. And that's basically precisely what we want to test in as part of this study related to CBD and autism. So we will take Dr. Mority's organoids and neurons that, and again, these sort of very, each individual patient in the study will have organoids derived from their own skin cells, basically. And we are going to carry out this analysis and look at this ratio in the individual patients. And depending on what that analysis shows, both positive and negative results will actually teach us something about the underlying physiology. How is information changing? How is it different in these patient-derived autistic networks? The ultimate dream, if you will be to be able to almost predict which one of these patients will respond clinically. Now that's sort of the pipe dream. That's the ultimate objective. But if we understand enough in terms of network signaling, what's happening in these organoid models and in these neurons from these patients, we may be able to actually inform that both before and after sort of the treatment with CBD.
That was great. Thank you.